My name is Jonathan Irish, and I'm an assistant professor in the Department of Cancer Biology here at Vanderbilt University. So my lab is interested in how signaling controls cellular interactions, both in healthy development and in cancer. And our lab's toolkit really focuses on single cell tools. So what we want to do is peer into the cells and understand their signaling networks, get at how the cells think, and maybe change how they think by stimulating them particular ways or by treating them with compounds, and control the outcome of cell signaling interactions. There were a lot of things that attracted me to Vanderbilt, and so one of the key things is the amazing instruments and shared resources at Vanderbilt. So single cell tools, flow cytometry is really essential to my research, and the flow cytometry facilities are phenomenal at Vanderbilt. They brought in this new technology called CyTOF, and we were one of the 15th institutions in the world to get CyTOF, and it was very, very new at the time. It was the Generation 1 instrument, and it was just fantastic that Vanderbilt made that investment in the technology, but also made it a shared resource that's available to everybody at Vanderbilt. And so Vanderbilt is really one of the top institutions for marrying computational biology with basic research. And that really attracted me because there's not only the interdisciplinary graduate program, which recruits some of the top graduate talent for bench science, but there's a quantitative and chemical biology program that brings in computational and quantitative students. And those students all interact together in the same graduate program, and I can, as a PI, draw on all of those students. Recently, one of our big successes in the last year is we created a computational tool that's a first step towards teaching computers to identify different types of cells, to identify cancer cells and different populations of immune cells. And now we might take a new sample that might be blood from a healthy person or blood from a cancer patient and ask, can the computer identify cancer cells within that sample? Or could it even suggest a treatment that would be matched well to the type of cells that we're seeing within that sample? The excitement of revealing new types of cells and the power of understanding how to better treat a patient based on the biology of their disease, based on the biology that we see in their single cells, is really what kind of keeps me going every day and animates our lab.